This is a video showing the mechanism behind neurotransmitter release, the 4 categories of neurotransmitters, different kinds of receptors, and termination of signaling. This is the presynaptic neuron on the left and the postsynaptic dendrite on the right. The synaptic cleft is in the middle and is about 20 to 50 nanometers in length. Neurotransmitters are molecules in the presynaptic neuron that affect the postsynaptic neuron. There are 4 different categories: amino acids, peptides, acetylcholine, and the catecholamines. Amino acid neurotransmitters include GABA, glycine, and glutamate. For example, glycine is synthesized from serine by hydroxymethyltransferase, and it is put into vesicles by vias. Likewise, GABA and glutamate are also made from precursors. Peptide neurotransmitters, which are encoded for by a gene, include the enkephalins and dynorphins. These are made in the soma and diffused down the axon by vesicular transport. Acetylcholine formation requires the enzyme choline acetyltransferase. Acetylcholine is synthesized in the axon terminal and is placed into vesicles via a transporter. Acetylcholine is the primary neurotransmitter at the neuromuscular junction. Lastly, the catecholamines include dopamine, norepinephrine, and epinephrine. Tyrosine is the precursor to all of these. Neurons that synthesize these are involved in attention, movement, and mood. So when an action potential that is generated reaches the axon terminal, voltage-gated calcium channels open, and this allows calcium to rush in because of calcium's chemical and electrical motivation to do so. When this happens, this causes vesicle docking, which is shown right here. And this also leads to exocytosis, which releases the neurotransmitter into the synaptic cleft. The neurotransmitters can then bind to different receptors on the postsynaptic neuron. The first kind is known as an ion channel receptor. When a neurotransmitter binds to this, there's a conformational change in the receptor, and this leads to ions coming into the postsynaptic cell, such as sodium is the most common one. And then that can lead to depolarization and an action potential in the postsynaptic cell. G-protein coupled receptors have more diverse effects on the postsynaptic cell. For example, when a neurotransmitter such as dopamine binds to a G-protein coupled receptor, a secondary messenger molecule such as cyclic AMP, which is shown here bound to the receptor, can lead to a cascade of intracellular events in the postsynaptic neuron. This can also actually lead to the opening of other channels. A disadvantage of G-protein coupled receptors is that they are much slower, and because their mechanism often involves more steps. Eventually, it will be important to clear the synapse so that the neurotransmitters do not continuously bind to their receptors beyond what is helpful. Also, we do not want desensitization to occur. There are a few ways that a signal could terminate. First, the molecules could diffuse out of the space in the synaptic cleft. Secondly, amino acids, amines, and serotonin can be taken up into the presynaptic cell to be used for later. How that happens is once there's a certain amount of neurotransmitters in this space, they will actually be able to bind to to receptors on the presynaptic neuron, and it acts as some sort of negative feedback inhibition. So once there's a certain amount here in this space, it will bind and then be taken back up into the presynaptic cell. Third, there are actually transporters on neighboring glial cells, such as astrocytes, shown here, that cause neurotransmitters to be taken up and degraded. Lastly, enzymes can directly degrade neurotransmitters in the synapse. For example, monoamine oxidase is able to break down norepinephrine and epinephrine so that their component parts can be used for later neurotransmitter production in the neuron. Thank you for watching this video.